Okay, I think in the interest of time, we'll probably start our innovation showcase, our presentation today. So thank you to everybody for attending. It's really great to be here today and see a lot of familiar faces and have this room so well attended with our friends and colleagues and a lot of hungry people, it looks like, so this is great. My name is Coby Carlson, and I'm presenting today with my colleague Chris McMahon. And the title of our presentation is Human iPSC-derived Midbrain Dopaminergic Neurons for Parkinson's Disease Modeling and Cell Therapy. So I know you can all read that, but here we go. And so stem cells, iPSC technology, that's the reason all of these scientists are here in Boston today, right? So I'm not going to belabor how all of this works up here. I'll just point out a few important details, highlight a few things, and really just these three areas. And one is reprogramming. You can take adult cells and reprogram them back to an induced pluripotent cell line. The second is genome engineering. Everyone knows a lot about this. At the IPS level, you can edit a genome to knock in or knock out mutations, correct disease mutations, etc. And number three, differentiation. Taking an IPS line and turning it into one of these specialized human cell types. So, these three core competencies are really what our company in Madison, Wisconsin, Cellular Dynamics, was founded on. And, as this field has grown over the past 10 years or so, I would say that, CDI has been there along the way to help demonstrate, this transformative potential of iPSC technology and how really you can use it and how it's enabling for toxicology, drug discovery, and regenerative medicine. And I think it was the technology and the people and the work that happened at CDI that really grabbed the attention of a large Japanese corporation called Fujifilm. And so just giving a little bit of background, in case you did not know, about 2 years ago, Fujifilm acquired CDI in the spring of 2015. And, this gave some long-term stability and some direction for our company. And just this last fall, there were some other changes that were made where Fujifilm and CDI kind of realigned into two separate but equally important business units. One to serve the life sciences and tools space that we're kind of already operating in, and the other one really focused on cell therapy. And so it's based on this new realignment of our company is how we're setting up our talk today, right? So the first part is going to be given by me, really focused on disease modeling and using iPS cells for disease modeling in the life sciences area, in vitro tools. And then Chris will deliver his presentation on our cell therapy program for Parkinson's disease, okay? Easy enough. Okay, so CDI's life sciences portfolio, we make and manufacture human iPSC-derived cell types. So we've got the schematic here showing all the different ones that we make. So there's, at the moment, 14 different cell types, all cryopreserved, frozen cells in a vial, shipped anywhere around the world, thaw, plate, use, go, run your experiments. And so, it all started with cardiomyocytes for us, but we've really expanded, since then. And the focus of the talk today is really going to be on dopaminergic neurons. And, this cell type that's important, of course, for Parkinson's disease, that's one of the first cell types that degenerates. Okay? So we've presented, we've probably started working on this cell type about 4 or 5 years ago. And it's a long process to really optimize differentiation protocols and industrialize them for commercial use. But just like everybody else that's working with iPSC, we take our cues based on the literature. And so we developed a protocol for differentiating human midbrain dopaminergic neurons, based on published literature. And then we licensed a protocol from Studer Lab and Memorial Sloan Kettering. So what you can see here is, just a schematic, general schematic of of the process over time, describing some of the different stages of the differentiation, and having, some days in the process indicated and some images of what the cells look like along the way. And, when we brought in the protocol and started working on it, there were certain things that we had to do to, add some additional tweaks for the differentiation to make it specific for CDI to be able to make a larger scale, and just examples of some of the things we do, optimizing the concentrations of certain compounds or timing, et cetera. And it was these tweaks and workflow improvements that got us to a really robust 40-day protocol that we have excellent reproducibility. This cell type is really consistent, and we do it on a scale now that each batch run yields about 4 billion cells. And so this is a picture of a 50 mil Falcon tube filled with about 4 billion neurons. So that's what it looks like. And this cell type, dopaminergic neurons, are a percentage, 1% I think, of all the cells in the brain, neurons in the brain. There's about 400, 500,000. So this is a lot of cells that you would get from many brains. And importantly, we have optimized the cryopreservation protocol so we can use these cells for these life science in vitro tools use. Okay, so again, we've also presented in the past, at the ISSCR meeting and other places, the characterization of the cells. So I'm not going to go over all the specifics on this, but we have lots of literature that's available and things we can talk about even more offline. But we have high neuron purity, high markers for dopaminergic neurons. We have lots of high viability from at thaw when the cells are taken out of cryopreservation. We've characterized the cells for their gene expression and looking at dopaminergic identification and all the different neuronal subtypes at different days post-thaw. And then we've looked at how the cells are in culture, the phenotype, the morphology, and stain for the different markers. So, again, we have this is about as much as I'm going to say about the characterization of these cells. There's a lot more that Chris has coming up too. But we've also taken them and we've developed various applications with the cells. So not only is it one thing to make a cell that has the right markers that look, smell, and feel like a dopaminergic neuron, but they definitely need to have the functionality. And here are just some examples, sort of graphical abstracts of the cells tested on a multi-electrode array or in calcium oscillation assays, looking at cyclic AMP signaling with dopamine receptor agonists and cyclic AMP assays, mitochondrial toxicity or autophagy. So we have lots of different things. So we definitely have been able to use these cells in multiple formats. But, like I said, the focus of my part of the talk today is really talking about disease modeling and this, concept disease in a dish. And, iPSC technology now enables you to take samples from a healthy donor or a donor with a genetic disease and reprogram their adult cells back to the iPS state and differentiate them into a cell type that you want to study, and then run functional assays and phenotypic assays and compare how that works. So there's three sort of ways to do disease modeling. We call it innate, which is how I just described, comparing two different donors. Engineered would be where you have a healthy donor, a normal iPS line, make the iPS cells, and then engineer at that iPS level to put in a disease. Or conversely, you could take the genetic disease and correct it and make the isogenic control. But anyway, you've got, you can imagine what's going on here. And then the induced state, where a healthy donor, you put the cells under a condition of stress or et cetera, and induce the disease. So these are our three sort of buckets for disease modeling, and really going to focus today on the engineered model. And this slide goes into a little bit more detail about, making and testing a cell model for Parkinson's. And just sort of tilted this around and showed, but basically our starting material here is an apparently healthy, normal male iPS line. So we were programmed to generate this iPS line. And then at this stage, genome engineering of the iPS line to introduce the A53 mutation into the alpha-synuclein gene. So we have then isogenic iPS lines. This is a heterozygous mutation. And then both of those lines together were differentiated in parallel to make dopaminergic neurons, just the same process that we just illustrated for the eye-cell DopaNeurons. And we've done this, multiple times, multiple batches, and multiple lots have been made and tested. And we'll show some of, examples of the functional and phenotypic analysis, qPCR, ICC staining. We've got some metabolism and calcium and MEA assays to show. And, some things that I'll just address right now that we still want to understand, and maybe we can talk about at the end, but we would definitely want to understand, how these changes in this mutation affect the cell death or the viability of the cells. Do they release less dopamine, and how is the alpha-synuclein aggregation affected? So these are things that we're working on, but don't have data to present on today. So the first thing here is qPCR data. So this is showing the differential gene expression between the mutant and the wild-type cells. So all of these are normalized qPCR data, where black is the wild-type and gray is the A53T mutation. And what we're looking at are certain markers, and then here in blue you can see that these ones are actually the enzymes that are involved in the biosynthesis of dopamine. And so tyrosine gets converted to dopa by tyrosine hydroxylase, and further converted by dopamine decarboxylase into dopamine. And then, this enzyme, COMT, catabolizes dopamine and takes it away and turns it into this molecule. So why is this important? Well, even though I said we didn't measure the concentration of dopamine that's released from this, these data suggest that if you have decreased TH, as shown here, and decreased DDC, you're not likely making as much dopamine. Similarly, if you have increased COMT, any dopamine that might have been made is also likely converted to something different. So it's suggesting that the biosynthetic levels of dopamine, should be decreased. Okay, so for staining, there's, again, nothing quantitative on this slide, just showing you that, alpha-synuclein A53T mutants have strong staining for this marker. And there's a lot of different antibodies for this, and, we've screened a variety of different ones, and we found that this one was one of the best, and it just indicated, perhaps that there's certainly alpha-synuclein present, and we're working on ways to quantify this to show that you could have increased levels of alpha-synuclein or maybe even aggregated. Okay? So the next bit of data is using the Seahorse platform, looking at the bioenergetics, the mitochondrial respiration profile, and what is shown here that on the oxidative consumption rate is on the y-axis, and it's normalized to the number of cells, and this is over time. And if you're familiar with this assay, this is a really it's a kit-based experiment. It's standard, or adding in different mitochondrial insults along the way. But what you can see here is that the profile for the wild type is in green, and the responses look relatively as expected, normal as expected. But in blue, the baseline for the A53T actually starts a little bit higher, but most interestingly, when you add FCCP, the increase in the mitochondrial respiration is much higher. The capacity seems to almost double what it is for the wild type. And so, these things, whatever the reason for this is, it's certainly different, right? You've observed something different in vitro between these two. Okay, and so we also, when we manufacture any type of neuron, we want to understand how those cells operate and connect and fire action potentials in a dish. And one way to do that is to use multi-electrode array. So this is an electrophysiological readout where cells are placed in a dish that have electrodes on the bottom of the wells, and they can measure the voltage that comes from the neurons firing. And so what you're seeing here I'll walk you through this. So on the left are the wild type, on the right here are the alpha-synuclein A53T mutants. And this is a graph that we call a velocity graph. And down below it are raster plots. And each of these lines down here are electrodes. And going across in time, every black mark is a tick. and every tick means it's an action potential. So what you can see here is that at this point in time, at this culture, that there's lots of, this is also quantified on the side here, number of active electrodes. There's a high activity here. That all the action potentials are organized and firing and bursting in a synchronous fashion. And you can see here that there's an intensity of about 400 or 500. The alpha-synuclein mutant, though, on the other hand, plated at the same time, cultured under the same conditions, put in the same plate, have really a different profile. You can see that the bursting pattern is unique. So the time between bursts, the burst per minute, is definitely less, much more spaced out, and that's quantified here. And the intensity of these bursts, the y-axis is highlighted here to 2,000, that the intensity of those bursts are much, much greater. So just to review that, A53 T cells have fewer bursts per minute, but the intensity of those are really about 5 times greater. So this is a really unique phenotype. And then the last thing that we're showing here is that this is a calcium oscillation assay that's measured on the Hamamatsu FDSS. And what this is that the cells are plated and measured at day 7 and day 14 after thaw. And it takes usually about 14 days for these cells to network and start firing synchronously. So if you're looking here at the wild type in this group of cells, you can see that some oscillations might start happening, but they're really not connected and synchronous and rhythmic yet. On the other hand, A53T mutant at day 7 has high amplitude bursts, very regular, very pronounced, and happening early in culture. You wait one more week, the patterns start to look a little bit more similar. The wild type catches up, and still the intensity of these bursts are different. So again, generating a unique phenotype from this isogenic disease model. Okay, so that's just running you through that data right there quickly, modeling disease using this, familial PD mutation engineered in the iPS line to make these isogenic pairs, testing them in parallel across a sample of different assays shown here. And, clearly these are distinct phenotypic differences that have been observed and repeated and can really be useful for trying to understand the mechanism of what's going on with this particular monogenic mutation. All right. So last couple slides here, what's next? I think that what we're hoping to do and what the plan is to make more dopaminergic neurons from more donors so we can understand and offer tools to study Parkinson's disease even more broadly. So recently we announced that we've connected with the Michael J. Fox Foundation, and we're working with them to reprogram 85 different iPSC lines. It's part of their Parkinson's Progression Markers Initiative, this PPMI. And these different donors with and without Parkinson's disease, and we're going to be using these and make them available, expanding the access to the research tools for everybody to get to these, and then start making the DopaNeurons from those and running these same sorts of experiments that I just showed you to help illustrate that, or at least even also developing the protocols to pass on to other people so you could be running those same experiments in your own lab. Okay? So I think that's my last slide. This is sort of the halftime and intermission as I let Chris stand up here. Just wanted to put this up here. I always get asked all the time, why iPSC? Why would you use iPS cells to study disease? I can get lots of other cells for cheaper and easier, and I don't think I really need to explain the point of this slide, but you can see the list of different cell lines here on the left, and how normal they may or may not be based on the number of chromosomes they have on the right. So that's just a little, food for thought, and thanks a lot for your time. I'll answer any questions at the end, and I'll go ahead and let Chris jump up and finish out the talk. Thanks.
Great. Well, thanks everyone for coming. My name's Chris McMahon. I'm the director of the cell therapy program for Parkinson's disease at CDI. And I'll tell you a little bit about our therapeutics approach that we're taking, and then go into some detail about the Parkinson's program. So I'm preaching to the choir here, but we feel that the time is right for iPS-derived cell therapies. As a company, when we were founded more than 10 years ago, the business model was to reprogram iPS cells, to differentiate those into a number of different cell types, and then to make those available to the research and pharma markets. But the long-term plan was always, the assumption was always that eventually we would want to pivot into cell therapy when the time was right. And that's where we are now for a variety of reasons. First of all, the science is there, as you know, and you've been hearing this week. The differentiation protocols are good enough and look promising enough to proceed to trials. And also, as a company, we got really good at making lots of cells in large batches and good purity and excellent quality systems. And with the acquisition of Fuji, this is a focus area for them, and have given us the backing to do this. So there are several different paradigms for the way that iPS cells can be used in cell therapy. And again, I'll go through this quickly because everyone in the room knows. But you can take blood or cells from a patient, reprogram those into iPS cells, differentiate those into the cell of interest, and they can be put back in an autologous type of therapy back into the same patient. That is excellent as far as having very low risk for graft rejection, but it's also very laborious, time-consuming, etc. As with embryonic stem cells, cells can be differentiated. The stem cells can be differentiated into the cell type and put in an allogeneic into any patient, but this has a much higher risk for tissue rejection and for graft rejection, and in almost all cases, the patients have to be heavily immunosuppressed. We're taking a middle road, which is the HLA matching, so we're generating banks of iPS cells from HLA people with high-frequency HLA haplotypes and generating lines from those. And so the way that works is if you take donors that are homozygous for very common HLA haplotypes combinations, take blood from them and generate iPS lines from those, the cells from those banks are at least a good match or a partial match with anyone in the population that's homozygous or heterozygous for that same HLA haplotype. And so this is well underway. We've started what we call phase 1 here of the top 5 most common HLA haplotypes in the US population. We've reprogrammed those. 3 of those have been made into GMP master cell banks, and that covers about 35% of the US population. Underway is phase 2, where we are going to cover the top 12, which gets us over 50% of the population. And the eventual goal, you would have to do, you get this diminishing returns as you do less and less common haplotypes. So it takes 183 iPS lines to cover more than 95% of the population. So we're in the planning and assessing stages of that, and also looking in a more global way of looking at common HLA haplotype combinations in other parts of the world. These are not just for our internal use. The GMP iPS cell lines from these haplotypes are available to CDI partners for other clinical programs. So as far as our pipeline, we have 4 different programs going forward. So there's an ocular program using RPEs. This is fairly well advanced. It's a project that's being done in collaboration with the NEI to treat macular degeneration. And then we also have a photoreceptor program going that's more in the discovery phases. Cardiac, treating heart failure with cardiomyocytes. The neurodegenerative Parkinson's using midbrain dopaminergic neurons, as I'll go into detail about. And then also in discovery phase in oncology, so using iPS cell-derived CAR T and CAR NK cells for treatment of tumors. So Parkinson's. It's a terrible degenerative disease. It's the second most common neurodegenerative disease, with over 1 million people in the US being diagnosed. And this number just goes up, the frequency of the population as age goes up, and of course this is going to be more and more of a problem as our populations around the world age. The mechanism is well described in the earlier stages of the disease: loss of A9 midbrain dopamine neurons that innervate the striatum, and This leads to a loss of motor control and other motor symptoms, tremors, rigidity, and as the disease progresses and additional cell types are lost, this goes on to dementia, depression, and other types of symptoms. There are several good treatments. L-Dopa to replace dopamine in the striatum works well, but it works less and less well over time with side effects, dyskinesias, and so forth. And there's also deep brain stimulation, which works well for some patients, but again, works less well over time and is fairly invasive with electrodes implanted in a pacemaker. It is a good candidate for cell therapies. You can imagine being a very specific cell type that's lost, and there's quite a long track record of implantation using fetal tissue grafts. And going into some more detail here, back in the late 80s and through the 90s, a whole series of clinical trials was done using fetal midbrain engraftment, and the results were all over the place. Early open label trials gave fairly good results. Certainly in some patients they had an almost full recovery for over a decade, others less well. But then when it moved on to larger placebo double-blind trials, the results weren't nearly as good. If you looked at subsets, especially younger, earlier onset patients, the results were significantly better than controls. But looking at the populations as a whole, it did not give significant results. And more importantly, there were serious side effects, graft-induced kinesias in many patients, and that's really what shut down this approach for more than a decade. It's being readdressed now in the Trans-Euro trial happening in Europe. It's doing a retrospective meta-analysis of what happened before, deciding what worked, what didn't work, and coming up with a consensus approach for how to do all aspects of the trial, including dissection, the cell prep, the patient selection, the method of immunosuppression, how the surgery was done, etc. So making the best guesses at that, and they've done, I think, 10 patients now, and the results of that are eagerly awaited. But that's really seen as a stepping stone to stem cell-derived treatments, and those are in progress by many groups around the world, and they're looking at stem cell-derived treatments anywhere from neural stem cells to embryonic stem cells differentiated into midbrain Dopaneurons or progenitors or iPS-derived. And the evidence for that working is quite good, not only in those fetal trials, but in preclinical studies, so using allogeneic Dopaneurons and taking those and engrafting. As you can see the graft here into the midbrain of a rat. The mode of action is multifold. There's the ability of the cells to engraft long-term into the striatum. These are the nuclei, just showing the cells there, and they persist indefinitely. They innervate throughout the striatum. This is Th-positive cells, and then they are able to integrate into the host circuitry and release dopamine in a semi-physiological fashion. So when we looked at this, we looked at our current process and said, okay, how are we going to transition this over to a cell therapy type process? And so looking at the differentiation of the cells, we start with the iPS cells, drive it through specification and maturation. But you can, our iCell product is a fairly mature postmitotic dopamine neuron, and that could be the candidate, or it could be drawn from different places in the process to look at progenitors or somewhere in between. There's also the question of purification. We use a purification step to make our iCell DopaNeurons, and so there's a question of whether that's necessary, and if so, at what stage do you do it? And then there's another number of other hurdles to get to something that's appropriate for clinical use. So of course, transitioning away from research-grade reagents, developing a new purification approach if that's needed. We use a genetic modification and a drug selection of our iCell product, and that's not appropriate for the clinic. And then also, I described the HLA superdonor approach. Then you really need a differentiation process that's robust enough that it's going to work across any number of iPS lines, because you have that whole HLA superdonor bank, and you need to be able to reproducibly make cells of high purity from any line, not just one line. So addressing some of these one by one, the transition away from the research grade reagents, the iCell DopaNeurons use a single IPS line, a CDI internal line, and we have transitioned over then to the GMP grade HLA superdonor lines that I described earlier. There's the use of extracellular matrix proteins that the cells are grown on. In the iCell process, they're of animal origin, and we've switched over to using recombinant human protein made in a human expression system. So that works great. The growth factors in many cases are xeno or the expression systems or non-human, and so those are all transitioned over to recombinant human and non-animal expression systems. Median buffer, wherever possible, transitioning over to cell therapy grade reagents, looking at the sourcing of them and everything else. And we're surprised to find that some of the materials used in the differentiation process have unacceptable levels of endotoxin in them. So we've had to transition over to ones that are endotoxin free. So the purification, as I described, we use a genetic and drug selection for the iCell process, and what that results in is a very clean, this is a neuron gating here showing MAP2 positive and no Nestin positive cells. If you don't do that sort of selection, you often get outgrowth during the differentiation process of these immature glial lineage type cells, and that is not only a purity problem but a potential safety problem. So we've had to come up with a new purification approach, and at the end of the day what this yields is something with very similar purity as we see with the iCell product. The cells can be cultured long-term post-thaw. This is about a month post-thaw with no outgrowth of other cells. So the challenge of making a more robust process that will work across IPS lines, we found that we are using small molecules and growth factors to neuralize, generate floor plate, and specify a caudal midbrain. And if you take that optimized process, it's optimized for the iCell line, you get, this is an in process, but showing very good progenitor patterning, co-expression of FOXA2 and LMX1. But if you take another IPS line and put it through that same procedure, you get very poor patterning. And so to encapsulate a great deal of work, we did a lot of large matrix experiments to optimize not just what molecules were used, but the timing of giving them the concentration, and then in addition other process standardization methods, in particular the stem cell culture, working with working cell lines to make the process more robust and efficient, and that ended up with a defined process. This is an example. These are 3 of the HLA superdonor lines, and this is again looking at the progenitor patterning, very good patterning, all using the same differentiation protocol to get to the end of the process and get pure neurons captured, shown here in the flow. Good expression of FOXA2, which is a good marker for the midbrain, the floor plate lineage, and TH expression on the majority of the cells. Took that a step further in collaboration with Deborah Hall at Rush University, where she obtained blood from 4 Parkinson's patients. We reprogrammed into IPS lines using GMP-compatible methods, and then differentiated them using the new methods that I described, and looked at the success rate basically. So it was 3 different donors, 3 IPS lines from each donor, and the progenitor formation, this is all in shorthand, was very good for all of them, and then taking them out to the end of the process, the neuron purity was excellent for all the lines, and in all but 2 cases, good midbrain and dopamine neuron patterning. And so it was a good success rate here. 7 out of 9 of the lines looked very good, and more importantly, 3 out of 3 donors, if you do 3 lines each, it's successful. So this is our current process. We're starting with working cell banks, GMP grade IPS cells, running them through the patterning and specification process. We're focusing right now on the more mature dopamine neurons that are similar to the iCell DopaNeurons, using the purification process, cryopreserved, And that's what we have most of our data on, and we've done our pilot clinical studies on. But we're also doing range finding experiments, drawing cells from different parts, different stages of the process to see in vivo how well they match up. And that's important, but those are long-term experiments. And we're able to cryopreserve, even with these at the end of the process, large batches, very similar to our iCell product, where we can make 200 or 300 vials of what is probably a clinical dose of about 10 million cells. So we've done characterization of the cells made using the new process. So the reason for that are twofold, looking at batch-to-batch repeatability, and also comparing to the iCell DopaNeuron cells, which is what we've done the most characterization on. Want to make sure we're making the same thing. That all looks quite good. I'm not going to go through every gene here, but looking at midbrain patterning, classic dopaminergic neuron genes, and also importantly, making sure there's not strong expression of midbrain, of forebrain markers and markers for serotonergic neurons, motor neurons, and other unwanted cell types. Also comparing, this is just ICC, comparing our the PD therapy cells to iCell DopaNeurons. This is again FOXA2 and LMX. The expression is retained all the way through the differentiation process to the end, and so it looks very similar and is high expression on all the cells, co-expression on the cells, and expression of TH in culture and FOXA2. This is just 1 day post-thaw. You put them down and you can see they start extending neurites right away. The activity of the cell is also good, looking at functionality, MEA, electrophysiology. We haven't done a ton of this, but the cells are very active. You can see firing across all the electrodes, and over time in culture they get this rhythmic bursting frequency, very similar between the two cell preparations. And importantly, they release dopamine. So in culturing for 2 weeks and using a competitive ELISA assay, you can see that they secrete dopamine at least as well as the iCell product. Just a couple slides about QC assays. So we're developing a long list of QC assays that would be used to ensure the purity and safety of these cells. Many of these assays were developed as part of the iCell DopaNeuron product development, so looking at the purity in process of the progenitors, the final neuron purity, midbrain specification, dopamine neuron purity, all using flow cytometry assays, and other things looking at sterility and karyotype and all that. But we've had to develop a number of new assays specific for the therapy program, and they're almost entirely focused on safety. So residual iPS cells is critically important, and looking at other off-target cell types, doing better characterization of anything that's left over that's not a dopamine neuron. Proliferative cells, since we're looking at a post-mitotic candidate, we can go in and see how many cells are proliferating and how closely does that correlate to safety in vivo experiments. And so for just a couple examples of QC assay development, wherever possible, we try and take something like ICC, which is a pretty picture, but it's pretty hard to put numbers on, and translate that over to a flow or other assay where you can get a nice hard number on it. So this is that FOXA2 LMX co-expression that I've shown you several times, converted to a flow assay. You can see a good batch here with strong expression of both markers, and a not so good batch here. It's not great separation, but you can see that the LMX expression, there's a problem. You can see it visually here, and that would be a fail in process. Proliferative assay, proliferative cell assay, I mentioned, tried a number of different markers, Ki67 and so forth, but looking at EDU incorporation is really nice, very bright, good signal to noise type assay. You can see it visually here. This is in process. You can see that there's not very many cells at that point that are incorporating EDU. This is using a commercial click at EDU assay. And at the end of the process, you get very good, it's very bright on the flow, very good separation. And for the mature cells, you can look at it, see the small population of cells that are still incorporating EDU, and as I mentioned, try and correlate that to in vivo data to correlate with safety. So I've done a number of pilot preclinical studies over the last couple of years, and this has almost entirely been done in collaboration with Jeff Cordauer, Dustin Wakeman at Rush University, and more recently again with Dustin at RexGen. The early studies were short-term engraftment studies. At that point, we're looking at the cryopreserved cells versus fresh cells, and reassuring ourselves that the cryopreserved cells right out of the vial engrafted, as well as freshly cultured cells. And that worked well. And then we're also looking at different variations on the differentiation process. There were longer-term 6-month and 9-month efficacy studies done. A number of these, again, looking at different process variations and also purification strategies. And then also a few short-term engraftment studies in primates using DopaNeurons, and then more recently using the PD therapy cells. So this is data from a paper that was just published this week in Stem Cell Reports. And this is looking at the short-term engraftment. This is actually a nigral graft. And you can see here the staining here of the human cells. The tip of the needle was there, and the innervation growing out. And importantly, you can see the directionality of the innervation going down the nigral striatal tract, so headed in the right direction. And we have additional studies now to look at longer-term type experiments, so we can look at really true A9 phenotype by looking at where the cells engraft. This is looking at the nuclei. So again, this is the graft. You can see that the cells stay put. They don't go anywhere. And importantly, staining with Ki67 in the graft, you rarely see next to nothing. Maybe you can find a cell, but there's just no proliferation going on there, and we haven't seen any sign of tumors in any of the animals that we've looked at, short-term or long-term. The efficiency of the engraftment is good, ranges between 10 and 25%, so a large number of the cells that we're putting in there survive long-term. So that's also good news. So then looking at the efficacy studies, this is the this is the model system that's most commonly used, and so 6-OHDA is injected to the forebrain bundle, and this damages the DA neuron terminals. There's retrograde degeneration, and then what you get in the end is a unilateral ablation of almost all of the DA neurons on one side of the brain. Those animals then show hemi-Parkinsonian behavioral deficits. They spin in a circle, and that can be exacerbated by giving them amphetamines or apomorphine to make them spin faster. And this can then be rescued. In the case of engraftment studies, you can put in the graft, and after it engrafts well enough throughout the striatum, you can hope to rescue those behavioral symptoms. And so this is some data from that same paper. The graft is in there, so there's a strong graft. There's innervation throughout the striatum. It's not, at this time point, or with these cells, it's not as robust as you see with the endogenous cells. But it is enough to manifest recovery in the system here. So those animals that have grafts show full recovery by 6 months. With the amphetamine rotations and a significant improvement of the apomorphine rotations. And we've repeated this more recently in nude rats, where, again, in that case, all the animals had successful grafts and all the animals recovered. Short-term studies done in primates. This is cyno macaques at 3 months. And again, this is the graft site. You can see good innervation from the graft. You can see a large number of Th-positive cells at the graft site. And then more recently, this is using now the PD therapy cells and done with RexGen. Similar study, short-term 3-month engraftment, and again, the human cells that have a form of strong graft and are innervating, and there's a large number of Th-positive cells in the graft. So that's where we're at. The HLA IPS banks, the stage 1 is complete. We're working with those most highest frequency HLA superdonor lines right now, but the eventual goal is to extend that out to more and more. We've completed all the transition to clinical grade type reagents. We're happy with the purification approach. And the process working across IPS lines. The scale-up is certainly good enough. As I mentioned, we can make 300 vials no problem, and probably double or triple that with no problem. that can get you through phase 1, phase 2 trials. Down the road, additional changes will have to be made, and that we're already doing testing on that to be able to scale up to larger and larger types of bioreactors. Preclinical studies, multiple promising studies now. These have all been exploratory pilot studies, and we're trying to reach finalize our process lockdown so that we can do definitive studies. And the clinical studies clinical trial design is really in the planning stages now, but we've gotten as far as forming a scientific advisory board and getting their input on how to proceed with this. So with that, I'll finish up. This is the gang in Madison, Wisconsin. I'd like to thank you for your attention, and Coby and I'd be happy to take any questions. Yes?
So the early work by Thomas Bjorkman and his group to identify the critical stage of development of these dopamine cells, they were able to find that in the rat, E13 to E14, E14 and a half is about the ideal.
Mm-hmm.
They took a cells earlier than that or later than that, maybe rapid wasn't as robust, but maybe functional effects.
Yes.
So I think they then took it to the clinical level where they look at, first trimester. So I guess the question you pose here is, for you, what is the best, cell type in the stage of development? Have you looked at the transcriptome at each stage of your development and then compared it with first trimester to see what the match would be?
We're doing that now based on recent papers that have come out to try and better define what the cell mixture is, because it is heterogeneous. It's not a pure dopamine neuron population, and also developmental stage and what's critical. And both Bjorklund and also Studer's group have done these things to try and define what's the right age of cell to use, and it makes sense, but I think there's some caveats, because a lot of times they make this conclusion that the older cells don't work well at all. But we've found, just from a very practical sense, that the handling of the cells is critical. If you mistreat the cells, or if you have low, the health of the cells is not good of a more mature cell type, which is very easy to do, then everything can go bad very quickly. So the health of more mature cells is critical, and if you try and keep that high, that you can get good results. But we are doing the more just head-to-head comparisons to make up our own mind on that.
And so what has Studer chosen with his clinical trial in terms of, what stage of development?
Well, I don't know with what he's done with those preclinical studies, but there was more mouse studies that were done earlier using reporters where they reached the conclusion that cells that had just left the cell cycle, they're kind of Neur1 positive, were the ideal for transplantation.
Great. 2 questions. First, for cell therapy cells, is the purification step also based on selection of the set? And if so, would that be a problem?
No, the purification does not rely on selection. It's a different purification approach.
Okay, so there is no genetic...
Right,
okay.
No modification.
Okay, so good. Second question for your preclinical studies. Do you try to use the progenitors as well, or in that case the just went directly for the final documentation?
The early studies were all using a more mature post-mitotic cell, but we have experiments in progress now where we're doing that kind of range finding. We did some early where we used the mature post-mitotic and a little bit earlier cells, and we didn't see any differences there, but now we're doing a more complete, across the differentiation process to get a more complete answer on that.
Okay. But do you think there will be a better environment or using those progenitors?
It is a potential safety problem, and the other groups are using progenitors, and they're having to struggle with that. In some cases, they do see tumors using progenitors, and other groups don't, and I think it has a lot to do with how good the patterning is up front. But that's why our early focus was on using this post-mitotic cells, because the safety profile is so good. You can just look and see how many dividing cells are in there, and you know that that's not going to be a problem for the most part.
And I'll just more general comment. Will these type of therapies need a suicidal gene approach as well, or do you think this will always be?
We haven't, we thought about it, but we've decided not to go forward with that. And that's also, I mean, Studer's group, the same thing. They had a lot of, I don't know if you saw Vivian's talk yesterday, but they had a lot of kind of fancy ideas on things that they were going to do. And I think down the road, these things could be great, but it's just too difficult to take that to the clinic in any kind of a speedy way.
Thank you.
Thanks. And more or less related what he asked you, but I was wondering of which cells are you to study? All the pool of final neurons differentiated?
I'm sorry, I didn't
The
catch.
final drug cells that you are injecting to the drug amount, whatever, are the whole population of the neurons that you get at the end of the differentiation?
Is it a pure population of neurons?
Or if you purify it perfectly to inject some?
The neuron purity is high in the cells we're looking at, but it is a heterogeneous mixture of cells. I mean, we think that most of them are dopamine neurons, but there are certainly other cell types in there.
You show us that there's a small population of proliferative cells, so even that it's a small population inside the brain could produce tumors or proliferate. And so, did you remove them, or did you purify structures before transplantation, or all of it?
It's kind of a mixture. We're definitely trying to focus on and eliminate the cells that we think are potentially dangerous, and there is pretty good literature that serotonergic cells, for instance, may contribute to the dyskinesias, graft-induced dyskinesias. So we definitely want to get them out. And for the other cells, we're less worried about it. We definitely want to characterize them. We think the FDA cares about that. But trying to get the purest cell population possible is the goal, but is probably not attainable at this point in time. I think.
Hi, thanks. Just a couple of quick questions on these cells for in vitro use. You are able to cryopreserve the fully mature neurons, not just the progenitors?
Well, I wouldn't call them fully mature, but they're post-mitotic, immature dopamine neurons.
You put them straight into assay plates, and they still need to go through maturation at that point?
You can, depending on what type of assay you're doing in vitro. You can mature them out, and you can carry them for months.
Okay. What's the percentage of pH positive cells that you've seen at that point? How high is your ratio?
Yeah, the staining is a little funny because you get this range of, it depends on your media and your assay conditions, of a large number of cells that are TH kind of mid-expression and a subsection that are high. So depending on how you define them, it's between 25 and 70%. So it's a wide range, I know, but that's the best we can do.
Thank you guys. So you showed really interesting data that you can go into the animals and identify the transient cells. As you begin to amass your bank of the HLA subtypes, do you have any plans to be able to identify the cells and if it was from a sample from a person how to see them, like Y chromosome or like building the bank to be able to do that?
Oh yeah, I mean, we should be able to use genetic tricks to be able to identify specifically the cells that are from the graft. Yep.
Thanks for the talk. I was wondering, out of the cells that survive post-transplantation, do you have an idea of what fraction are pH-positive dopaminergic neurons?
Yeah, in the studies, it's ranged between 10 and 25% of the cells that go in survive long-term.
Do you know what the rest are?
The rest of the
cells that are surviving.
Oh, I see. I think I got your question wrong. You're asking what percentage of the cells are TH positive?
Right. Or like dopaminergic neurons.
Right.
How are you characterizing them?
Yeah, there's Justin there. I mean, I think it was up to 50%, right? But it was 25 to 50%?
Yeah. It was interesting because actually the animal had the lowest survival rate had the highest TH percentage, which was 50%, and the animal with the highest survival rate had the lowest amount of TH. And so I think what we've learned from fetal grafting studies is that it's not necessarily about getting the highest number of cells to survive, it's about getting them to innervate. The innervate, you saw this variety, the innervation was extensive, and that's actually quantified in the paper, you can see. So it's quite impressive. So again, when you think about dosing and going to the FDA, they really want to know how many times, how many needle tracks you're going to put into a person and how many cells you're going to put in there to get a therapeutic effect. What we have right now is we think it's around, 1 million to 10 million, depending on who you're looking at, right, where you're going into. The composition of the graft itself, though, what we think those non-TH, mature TH positive cells are. So Ernest Arini's group recently published a paper in Cell, I believe it was, and they used the exact same ISL-DopaNeurons, the off-the-shelf product. So by the way, these cells that we used in that paper are cells you can buy, you can reproduce that study. The composition of those cells that they've already seen on in the Arini's group showed that it was sort of a developmental continuum. So there's still dopaminergic progenitors or these neuroblast type cells in there. So we think what's going on is the exact same thing in these grafts, and this is an experiment that is probably in the works or ongoing, just to then now take these markers that came out of that paper, these and now there's antibodies for, and to go in and stain those and see how that composition changes over time. Of course, remember this is a rodent thing, right? So this is not the human condition, and we know from fetal grafts it takes several years before those cells really mature, even from human to human, really mature, innervate, and have the most significant impact clinically. So it's a great question, and I think it's something that they're ready to answer. So.
Yeah, but it's difficult because they don't bin very neatly into it's this type of cell. You know, it's more like I don't want to say Franken cell, but you know, it kind of falls in between different cell types, and some of them are along developmental continuum, some are radioglial type cells, but type, they're not firmly defined. It's an unsatisfactory answer, but we'd love to know the real answer.
Thank you.
Some questions here. You mentioned in your talk about the preclinical stages of these Parkinson patients and community. How do we get involved with that process, and when do we anticipate to do those studies?
For patient selection, you mean?
Yes.
Well, that's a ways down the road, but it's definitely going to be through a large movement disorder clinic, and they'll recruit people. But there's a lot of decisions to be made as far as exclusion, inclusion criteria. We haven't even made a final decision on the first trial. We'll do the HLA matching or whether it will be unmatched. So that's, but we'll be working with Rush Medical and hopefully Michael J. Fox and different to recruit patients. I'm sorry, I didn't hear it.
Where's Rush Medical or Rush University?
Chicago, I'm sorry, yeah, in Chicago. One more question?
One more. Do your cells, your DopaNeurons express in GRAIL, and do you see any correlation between GRAIL expression and surviving in GRAIL?
Yes, and functionality as well. That's critical. The protocol has been published recently by Parmar's group, but we reached the same conclusion quite a while ago, not as elegantly as she did, but that the FGF8 was critical for the differentiation to really get the caudalization to just the right spot, and the GRAIL expression, among other things, was a key marker for successful cells. Yes, yep. Yeah, I cut that slide, but we ran the whole panel of predictive markers that Parmar did, and we see good expression of all of those. Well, thanks for coming, everybody.